Let's continue learning about transcription. So once an open promoter complex has formed, RNA polymerase is ready to initiate synthesis. RNA polymerase contains two nucleotide binding sites called the initiation site and the elongation site respectively. The initiation site prefers to bind purine nucleotide triphosphate, namely ATP and GTP, and one of these is usually the first nucleotide in the chain. Therefore, the first base on the DNA template that is transcribed is usually a thymine or a cytosine. The initiating nucleoside triphosphate binds to the enzyme in the open promoter complex and forms a hydrogen bond with the complementary DNA base. The elongation site is then filled with a nucleoside triphosphate that is selected strictly by its ability to form a hydrogen bond with the next base in the DNA strand. The two nucleotides are then joined together by a reaction. During the joining process, RNA polymerase moves along the DNA to the next base pair. As it moves, it separates the base pair at position +4 and allows the base pair at position -10 to come together again, thus maintaining the same stretch of 13 base pairs unwound, but now centered one base pair further along the DNA. The two RNA nucleotides that were linked together stay paired to their complementary DNA bases, but now a new unpaired base, the one at position +3, is positioned in the elongation site. Again, the elongation site is filled with a nucleoside triphosphate that is selected by its ability to form a hydrogen bond with the DNA base at position 3. This nucleotide is then joined to the two that had been linked previously, and RNA polymerase moves along to the next base pair. Repetition of the cycle of nucleoside triphosphate selection and joining, RNA polymerase motion, DNA base unpairing, and DNA base unannealing or re-annealing gradually elongates the RNA chain. So RNA polymerase binds to promoter, slides into place, and forms an open complex. ATP in initiation site binds to T on coding strand. A NTP is added to the elongation site and is covalently linked to the A. RNA polymerase moves over to the next DNA base. A NTP enters the elongation site and is covalently linked to the dinucleotide. Then movement of RNA polymerase continues. Note that the selection is made by pairing of the nucleoside triphosphate with only one of the two strands of the DNA. The DNA strands were purposefully unwound to allow the selection to occur. The strand that has bases that pair with the incoming RNA nucleotide is the template strand. Since the sequence of the RNA is complementary to the template strand, it will have the same sequence as the other strand of DNA. After several nucleotides between 4 and 8 are added to the growing RNA chain, three important changes occur. First, the nucleotide at the 5' end of the RNA chain, the first one to be incorporated, becomes unpaired from the base on the template strand to allow that base to pair again with its complement on the other DNA strand. Second, RNA polymerase changes its structure and loses the alpha subunit. Third, a protein aiding elongation called NUSA protein binds. The rest of the elongation process is carried out by the core enzyme NUSA complex. Each new nucleotide that is added at the 3' end of the growing RNA allows another nucleotide to be released from its binding to the template. Hence, as elongation continues, selection of 4 to 8 nucleotides at the 3' end of the newly made RNA is paired to the DNA template strand, while the rest of the RNA emerges from the RNA polymerase as a single strand. Then termination of RNA synthesis occurs at specific base sequences in the DNA molecule called terminators. Some termination sequences allow RNA polymerase to terminate elongation spontaneously. These sequences are called intrinsic terminators. Other terminators require the action of a protein called Rho. They are called Rho-dependent terminators. Intrinsic terminators have three characteristic features, very important features. First, there is an inverted repeat base sequence containing a central non-repeating segment. That is, the sequence in one DNA strand would read A, B, C, D, E, F, X, Y, Z, F, FEDCBA, in which A and A dash, B and B dash, and so on are complementary bases. The sequence is capable of intra-strand base pairing, forming a stem and loop configuration in the RNA transcript and possibly in the DNA strands. The second region is near the loop end of the putative stem, sometimes totally within the stem, and a sequence having a degree high G plus C content. A third region is a sequence of A T pairs which may begin in the putative stem that yields in the RNA a sequence of 6 to 8 uracil residues, often followed by an adenine. Rho-dependent terminators lack sequences with a stretch of adenine residues in the template strand. Rho acts by binding to special sequence on the nascent RNA and forcibly pulling the RNA away from its contact with the DNA in the transcription elongation complex with RNA polymerase. Rho derives its force from the hydrolysis of ATP molecules. The final step in the termination process is dissociation of RNA polymerase from the DNA. This comes about by the rebinding of eta subunit to the core RNA polymerase after the RNA is released. The released holoenzyme is free to search for a new promoter where it can start synthesis of a new RNA. Then next major topic, classes of RNA molecules. There are three major classes of RNA molecules: messenger RNA or mRNA, ribosomal RNA or rRNA, and transfer RNA or tRNA. All the synthesized from DNA-based sequences. They are all involved in protein synthesis, but each class of RNA has a different function. As we will see in this and next chapter, there are also significant differences between the structures and modes of synthesis of the RNA molecules of prokaryotes and eukaryotes, although the basic mechanism of their function are nearly the same. The greatest amount of information has been obtained from studies with bacteria and bacterial cell extracts, so that it is here that we begin. Transcription in eukaryotes and the structure and synthesis of eukaryotic RNA molecules are discussed in a later section. So, messenger RNA, we are going to learn about messenger RNA. The base sequence of a DNA molecule determines the amino acid sequence of every polypeptide chain in a cell, although amino acids have no special affinity for DNA. Thus, instead of a direct pairing between amino acids and DNA, a multi-step process is used, and the information contained in the DNA is converted to a form in which amino acids can be arranged in an order determined by the DNA base sequence. This process begins with the transcription of the base sequence of one of the DNA strands or the template strand into the base sequence of an RNA molecule, and it is from this molecule, messenger RNA, that the amino acid sequence is obtained by the protein synthesizing machinery of the cell. As we will see, the base sequence of the mRNA is read in groups of 3 bases. A group of 3 is called a codon, we know that. And from a start codon to a stop codon, with each codon corresponding either to one amino acid or a stop signal. Then a DNA segment corresponding to one polypeptide chain plus the start and stop signals is called a cistron. So what's a cistron? A DNA segment corresponding to one polypeptide chain plus the start and stop signals is called a cistron. And an mRNA encoding a single polypeptide is called monocistronic mRNA. It is very common for a bacterial mRNA to encode several different polypeptide chains. In this case, is called polycistronic mRNA molecule. In addition to cistrons and start and stop sequences for translation, other regions in mRNA are significant. For example, translation of the mRNA molecule, that is protein synthesis, seldom starts exactly at one end of the mRNA and proceeds to the other end. Instead, initiation of synthesis of the first polypeptide chain of a polycistronic mRNA may begin hundreds of nucleotides from the 5' terminus of the RNA. The section of non-translated RNA before the coding region is called a leader sequence. In some cases, the leader contains a regulatory region called the attenuator that determines the amount of expression of the gene. Untranslated sequences are found at both the 5' and the 3' termini. P5' to 3' OH, protein coding sequence of mRNA. A polycistronic mRNA molecule may contain intercistronic sequences or spaces, hundreds of bases long. An important characteristic of prokaryotic mRNA is its short lifetime. Usually within a few minutes after an mRNA molecule is synthesized, nuclease degradation occurs. Although this means that continuous synthesis of a particular protein requires ongoing synthesis of the corresponding mRNA molecule, rapid degradation of mRNA is nonetheless advantageous to bacteria whose environment and needs often fluctuate widely. Synthesis of a necessary protein can be regulated as needed simply by controlling transcription. When a particular protein is needed by a cell, the appropriate mRNA molecule will be made. However, when the protein is no longer needed, inhibition of the synthesis of the mRNA is sufficient to prevent wasted synthesis because the previously made mRNA molecule will soon be degraded. Then stable RNA, ribosomal RNA and transfer RNA. During the synthesis of proteins, genetic information is supplied by messenger RNA. RNA also plays other roles in protein synthesis. For example, proteins are synthesized on the surface of an RNA containing particle called a ribosome. During the synthesis of proteins, genetic information is supplied by messenger RNA. RNA also plays other roles in protein synthesis. For example, proteins are synthesized on the surface of an RNA containing particle called the ribosome. Those particles consist of three classes of ribosomal RNA or rRNA, which are stable molecules. Also, amino acids do not line up against the mRNA template independently during protein synthesis, but are aligned by means of a set of about 50 adapted RNA molecules called transfer RNA, also a stable species. Each tRNA molecule is capable of reading 3 adjacent mRNA base codon and placing the corresponding amino acid at a site on the ribosome at which a peptide bond is formed with an adjacent amino acid. Neither rRNA or tRNA is used as a template. The roles of ribosomes and other tRNA molecules will be explained in the following chapter. Here we are concerned only with their synthesis, which involve transcription from DNA and some transcription modifications. The synthesis of both rRNA and tRNA molecule is initiated at a promoter and completed at a terminator sequence, and it is respect with their synthesis is no different from that of mRNA. However, the following 3 properties of these molecules indicate that neither rRNA nor tRNA molecules are the primary transcripts or immediate products of transcription. The molecules are terminated by a 5' non-phosphate rather than the expected triphosphate found at the ends of all primary transcripts. Both rRNA and tRNA molecules are much smaller than the primary transcripts, the transcription units. All tRNA molecules contain bases other than A, G, C, and U, and thus unusual bases, as they are called, are not present in the original transcript. All of these molecular changes are made after transcription by a process called post-transcriptional modifications, or more commonly RNA processing. Both rRNA and tRNA molecules are excised from large primary transcripts. Often a single transcript contains the sequences for several different molecules. For example, different tRNA molecules are both tRNA and rRNA molecules. Formation of rRNA molecule is a result of fairly straightforward excision of single continuous sequence. However, formation of tRNA sequence molecules from primary transcript requires not only cleavage by several enzymes acting in a particular order, but also chemical modifications of various bases. An example of the production of a particular tRNA molecule is very important, and that indicates the complexity of the process. Then next topic, transcription in eukaryotes. Now we are going to learn this topic in detail. This is a very important topic. So we are going to start the major portion of this series. Most of the basic features of the transcription and the structure of mRNA in eukaryotes are similar to those in bacteria. However, there are 5 notable differences. What are the 5 notable differences? Eukaryotic cells contain 3 classes of nuclear RNA polymerase, and these are responsible for the synthesis of different classes of RNA. Many mRNA molecules are very long-lived. Both the 5' and 3' termini are modified. A complex structure called the cap is found at the 5' end, and along up to 250 nucleotide sequence of polyadenylate acid, poly A, is found at the 3' end. The mRNA molecule that is used as a template for protein synthesis is usually about 1/10 the size of a primary transcript. During mRNA processing, intervening sequences called introns are excised and the fragments are rejoined. All eukaryotic mRNA molecules are monocistronic. These points are very important. It shows a schematic diagram of a typical eukaryotic mRNA molecule and how it is produced. There is clearly more to the production of mRNA in eukaryotes than just transcription of a DNA. The stages of processing of E. coli tRNA gene transcript, the 5 stages are given like step 3 generates the 5' P end. This step is catalyzed by RNA ASP, which is a ribozyme, an unusual RNA enzyme. Step 4 generates the 3' OH end, the CAA end. In step 5, 6 bases, all in or near the loops of the tRNA molecule, are modified to form pseudouridine, 2-isopentyl adenosine, 2-IPA, 2-methyl guanosine, 2-mg, and 4-thyroidine. The continuous sequence that forms the final tRNA molecule is given in black. Initiation of transcription in eukaryotes is not well understood. A sequence TATAAAA analogous to the -10 consensus sequence in prokaryotes but centered at -29 is a part of many promoters. Other sequences called upstream activation sites and enhancers, which will be discussed in later, affect the efficiency of initiation. These are usually sites where certain proteins bind. These proteins, called transcription factors, generally increase the transcription from promoters that are within the vicinity of the sequence to which they bind. So in some cases, promoters are as far as 2000 and 3000 bases away from an enhancer site along the DNA. In eukaryotic cells, DNA is bound tightly to histone proteins in a structure called chromatin. The structure of chromatin makes transcription more complicated in eukaryotes than in prokaryotes, and evidence exists suggesting that chromatin structure is temporarily reconfigured in regions being transcribed, although how it is altered is far from clear. This is also a mechanism for terminating transcription in eukaryotic cells, but very little is known about the general properties of the sequence or the factors involved. Then 3 types of RNA polymerase function in eukaryotes. The 3 major classes of eukaryotic RNA polymerase are denoted 1, 2, and 3. They can be distinguished by the ions required for their activity, their optimal ionic strength, and their sensitivity to various inhibitory compounds. All are found in the eukaryotic nucleus. RNA polymerase are also found in mitochondria and chloroplasts. The location and products of the nuclear RNA polymerase are very important, like nucleoplasm tRNA, 5S RNA, etc. The biochemical reaction catalyzed by the eukaryotic RNA polymerase is the same as that catalyzed by E. coli RNA polymerase. The 5' terminus of a eukaryotic mRNA molecule carries a methylated guanosine derivative 7-methyl guanosine, 7-MEG, in an unusual 5' linkage to the 5' terminal nucleotide of the primary transcript. Occasionally, the sugars of the adjacent nucleotides are also methylated. Capping occurs shortly after initiation of synthesis of the mRNA, possibly before RNA polymerase 2 leaves the initiation site and proceeds all excision and splicing events. The biological significance of capping has not yet been unambiguously established, but it is believed that it is required for efficient protein synthesis. Capping may function to protect the mRNA from degradation by nucleases and to provide a feature for recognition by the protein synthesizing machinery. Most, but not all, animal mRNA molecules are terminated at the 3' end with a poly A tract, which is added to the primary mRNA by a nuclear enzyme, poly A polymerase. The adenylate residues are not added to the 3' terminus of the primary transcript. Transcription normally passes the site of addition of poly A, so that an endonucleocytic cleavage must occur before the poly A is added. A base sequence AAA 10 to 25 bases upstream from the poly A site is a component of the system for recognizing the site of poly A addition. Interestingly, some primary transcripts contain 2 or more sites at which poly A can be added. The differentially terminated mRNA molecules usually play different roles in life cycle of the particular organism. The length of the poly A segment can be from 50 to 250 nucleotides. The significance of the poly A terminal is unknown at present, but it is believed that it increases the stability of mRNA. A possible role in enhancement of mRNA translation has also been proposed. Some cellular mRNA molecules lack poly A, so its presence is not obligatory for successful translation. Next topic, eukaryotic primary transcripts contain intervening sequences or introns. Most of the primary transcripts of higher eukaryotics or organisms contain untranslated intervening sequences or introns that interrupt the coding sequence and are excised in the conversion of a primary transcript to mRNA. The amount of discarded RNA ranges from 50% to more than 90% of the primary transcript. The remaining segment exons are joined together to form the finished mRNA molecules. The excision of the introns and formation of the final mRNA molecule by joining these exons is called RNA splicing. What is that process? RNA splicing process. The number of introns per gene varies considerably and is not the same in all organisms for a given protein. Furthermore, within a particular gene, introns may have different sizes and are usually larger than exons. Splicing occurs in the nucleus and in some cases starts on an RNA that is in the process of being elongated by RNA polymerase, a nascent RNA. Usually, however, splicing is carried out after transcription and poly A addition are completed. The existence of splicing explains how the nucleus can contain an enormous number of different RNA molecules whose size distribution is very great. The precursor and partially processed molecules are called heterogeneous nuclear RNA because of the great diversity in size of the molecules. After processing is completed, the mature mRNA is transported to the cytoplasm to be translated. Removing an intron without altering the coding sequence of the resulting mRNA molecule requires great fidelity in the cleavage process. For example, a cutting error that displaces the cutting site by a single base would completely destroy the reading frame for protein synthesis of the bases in the mRNA. Fidelity is provided by a base sequence itself. In all genes observed with which splicing occurs, the splice sites of the primary transcript are marked by a sequence resembling in which PY is an appropriate X is the base. The underlined bases are the same for all observed introns, that is, they are conserved. Whereas the remainder is a consensus sequence in which there is some variation from one splice site to the next. In the nucleus, a precursor to mRNA molecule like hnRNA are complexed with specific proteins performing a ribonucleotide particle, RNP. In the course of the splicing reaction, primary transcripts are assembled into particles called hnRNP. The process of RNA splicing occurs at a separate structure called spliceosome. Spliceosomes actually form on the splice site through interactions with a number of small nuclear ribonucleoproteins called snRNPs. A key component of an snRNP is at least one small nuclear RNA or snRNA. Each snRNP has a separate function. The one with the 165 nucleotide UA snRNA binds to the consensus sequence at the 5' end of an intron, while that with the U5 snRNA aligns the two exon sequence that are to be linked together. These RNPs bring the two interactions or junctions into close proximity at the spliceosome, which has enzymatic activities that perform the intricate process of cutting out the intron sequences and joining the two exon segments. The existence of introns seems to be fairly wasteful, especially in view of the relative amount of introns and exon RNA. In a few cases, the presence of introns increase the coding capacity of a particular stretch of genes. In that alternative splicing pattern yield different mRNA molecules. This is common in DNA viruses, but not particularly frequent in cellular DNA, although several examples are known. Then several theories have been proposed to explain why genes from eukaryotic cells are interrupted by introns. Since introns are almost never found in the genes from bacteria, they are considered to be simpler forms of life. It is reasonable to suppose that gene interruptions were added during the course of evolution. Various steps of recombination allow segments of DNA to be exchanged between different places on a chromosome. Thus, an interruption of a gene could appear by this type of recombination, and as long as the cell had acquired the means of splicing RNA, the right kinds of interruption would yield a functional gene. Then an alternative idea that interrupted genes were present even in the earliest organisms and during the course of evolution were lost by bacteria in order to simplify their genomes and allow them to duplicate as rapidly as possible. This alternative provides a plausible explanation for how genes for complex proteins might have evolved. Each exon could represent the coding sequence of an ancestral small protein, and rearrangement that occurred during the course of evolution could have brought the sequence together from various parts of the genome to form new genes. The use of an RNA splicing mechanism that would work to remove all introns would allow a range to be assembled and tried in new combinations without requiring precise recombination events to fuse parts and form a gene lacking interruptions. Examination of the structure of protein has revealed that many consist of several independently folded regions, each separated by a short polypeptide segment. Each individually folded region is called a domain. A protein with two very distinct domains is very important. This particular protein, a part of an antibody molecule, is encoded by a gene consisting of two large and two small exons. The two large exons code for two separate domains, suggested that each domain is derived from an ancestral protein. Now, next topic, means of studying intracellular RNA. Several procedures are used to study RNA metabolism in vivo. In most of the techniques, radioactive RNA is prepared by adding H3 labeled or 14C labeled uridine to the growing medium. However, this also produces radioactive DNA because uridine can be metabolized to cytosine and thymine. This is significant when studying RNA metabolism because when RNA is isolated from cells, it is usually contaminated with DNA. The contaminating DNA can be removed by treatment of the sample with pancreatic DNase. For this, enzyme degrades the DNA to monoribonucleotides and small oligonucleotides, which can be easily separated from the RNA. Most investigations of RNA in vivo are concerned with either of the presence, synthesis, or degradation of specific mRNA species, for example, the mRNA from a particular gene. In order to carry out such synthesis, it is necessary to distinguish the particular RNA molecule from all other RNA molecules. This is usually done by RNA-DNA hybridization. In this technique, DNA with a sequence complementary to the RNA molecule of interest is permanently fixed to a nitrocellulose filter. A filter is subsequently incubated with an extract containing radioactive RNA using conditions leading to base pairing between RNA and DNA strands and then washed repeatedly. RNA is not present in RNA-DNA hybrids is removed. The amount of radioactivity remaining on the filter is measure of the amount of particular RNA present in the extract. A variety of procedures are used to obtain DNA used in a hybridization experiment. In most useful experiments, a DNA segment containing a sequence complementary to RNA of interest is inserted by genetic engineering techniques into a piece of foreign DNA that has no sequence in common with the RNA and that is easily purified. In this case, the DNA sequence of interest is said to be cloned. Typical foreign DNA molecules are plasmids, phages, viral RNA. A DNA molecule carrying a cloned DNA sequence to be used in a hybridization experiment is called a probe. Similarly, an RNA molecule can be used in hybridization experiments, and in this instance, it is also referred to as a probe. The technique most commonly used is the Southern transfer or Southern blotting procedure, named after E. Southern, its developer. A method in which hybridization is performed with a large number of distinct DNA segments simultaneously. In this procedure, the total DNA of an organism is broken into discrete fragments by restriction enzymes. Which makes cuts in unique base sequences. Fragments are separated by gel electrophoresis, as in dideoxy base sequencing technique. DNA and hybridization is carried out on all of the fragments represented in a gel, and various techniques enable the positions of particular DNA segments or fragments to be identified. The location of hybridized radioactive RNA can then be matched with these fragments. The technique is performed. DNA is enzymatically fragmented and then electrophoresed through an agarose gel. Following electrophoresis, the gel is soaked in a denaturing solution, usually NaOH, so that all the DNA in the gel is converted to single-stranded DNA, which is prerequisite for the hybridization to be performed later. After the gel, which is typically in the form of a long flat sheet, is denatured, it is placed on a piece of nitrocellulose paper. The denatured DNA binds tightly to the nitrocellulose paper. DNA molecule do not diffuse very much, so if the gel and the nitrocellulose filter are in firm contact, the positions of the DNA molecule on the filter will be identical to their positions in the gel. The nitrocellulose filter is dried in vacuum to ensure that the DNA remains on the filter during the hybridization step. The dried filter is then moistened with a very small volume of solution of 32P labeled RNA probe, placed in a tight fitting plastic bag to prevent drying and held at a temperature suitable for renaturation, usually 65 degrees Celsius for 16 to 25 hours. The filter is then removed, washed to remove unbound radioactive molecules, dried and autoradiographed with X-ray film. The blackened position on the film indicates the locations of the DNA molecule whose DNA base sequences are complementary to the sequences of the added radioactive probe. Since the genes contained in each fragment on the gel usually are known, specific mRNA molecules in a mixture of RNA probes can be identified as corresponding to specific genes. The degree of blackening on the film is easily measured quantitatively and is usually proportional to the amount of RNA probe that has hybridized. Therefore, the amount of mRNA transcribed from each region of a DNA molecule can be measured with this method. Note that many different mRNA molecules can be studied simultaneously using this technique. The Southern transfer method also has many other uses, particularly in DNA hybridization analysis. Now we are going to look at a future practical application. As an alternative to conventional drug therapy as a method for combating viral infection, some researchers are investigating the use of antisense oligonucleotides to disrupt the metabolism of the virus. What are they? Antisense oligonucleotides to disrupt the metabolism of the virus. An antisense oligonucleotide is a nucleotide sequence that is complementary to either a single-stranded protein coding sense DNA segment or an mRNA protein synthesis template. In a living cell, an antisense oligonucleotide that has base sequence complementary to a gene will bind directly to the target gene and thereby prevent RNA polymerases from transcribing the gene. Similarly, an oligonucleotide that hydrogen bonds to a mRNA segment can disrupt its translation. Then short oligonucleotides consisting of as few as a dozen nucleotides have so far been demonstrated to be effective in disrupting either transcription or translation in test systems that consist of virus-infected cultured mammalian cells. A key advantage. So what is happening here? Short oligonucleotides consisting of as few as dozen nucleotides have so far been demonstrated to be effective in disrupting either transcription or translation in test systems. A key advantage of this method is that it leads itself to the so-called rational approach to drug design. That is, one candidate target nucleic acid sequences are known. Researchers can design appropriate oligonucleotides based on the simple Watson-Crick base pairing rule. Another advantage of this method is its specificity. The oligonucleotides should bind only to their target genes and therefore not interfere with other essential metabolic functions. Finally, the relative ease with which oligonucleotides can be synthesized provides for quick experimentation. Then disadvantages to the pioneering technology do, however, exist. A major one concerns drug delivery. Difficulties have been encountered with administering oligonucleotides to mammals. Frequently, these agents either do not penetrate target cells, or once within a cell, they are degraded by cellular RNAs. Nevertheless, novel delivery strategies which involve using lipid vesicles or carrier proteins and further refinements in the design of RNA resistant oligonucleotide structures are expected to alleviate those problems. Now we are going to have a quick summary of all the topics which we have covered in this sessions, and also with the help of previous videos. So RNA polymerase initiates transcription by first binding to promoter sequence on DNA. The bacterial enzyme does this by contrast between the gamma subunit of the holoenzyme and delta sigma subunit of the holoenzyme and sequence at 10 and 35 base pairs upstream from the start point. Eukaryotic RNA polymerase rely on recognition of the promoter by separate initiation factor. RNA synthesis is initiated by a reaction between two nucleotide triphosphates paired to residues +1 and +2 of the template strand. Chains are elongated by addition of nucleotides at the 3' end of the growing RNA at a rate of about 50 per second. Termination occurs spontaneously at certain DNA sequences called intrinsic terminators, or by action of a protein factor at a factor-dependent terminator sequence. In E. coli, that factor is called Rho, which acts at Rho-dependent terminators. The three major classes of RNAs are messenger RNA, mRNA, ribosomal RNA, rRNA, and transfer RNA or tRNA. rRNA and tRNA are derived from primary transcripts synthesized and previously described by various processing reactions that include cutting, trimming, and nucleotide modification. In prokaryotes, mRNA are usually primary transcripts, while in eukaryotes, mRNA are processed from precursor transcripts by addition of 5' caps and poly A tails, and usually by splicing together exon sequences. So with the help of these 3 videos, we have completed learning about transcription and some major aspects of this and some future applications, along with some experimental techniques, how to study intracellular RNA. So thank you for listening.